Katelyn Fritz, and her presentation is entitled Development of Genomic Resources for Increased Folate Content in Peanut Arachis hypogaea.
Thank you, Dr. Kimmerer. All right. Hi, everyone. Like you said, my name is Katelyn Fritz, and I'm looking at the development of genomic resources for increased folate content in peanut or Arachis hypogaea. So I'm going to start by introducing you all to what folate is. So folate is the term for tetrahydrofolates and their derivatives. So you can see on the figure on the right, we have the different species listed for folates, and they all just differ based on which R group is connected at the locations indicated above. So this is a very important molecule because it's used in single carbon transfer reactions. So these are things such as gene expression and lipid and protein synthesis. The folate biosynthesis pathway is supposed to be well conserved within plants, and it's an 11-step process over 3 subcellular compartments involving 9 specific enzymes. Within humans, it's very important that we get them from plants because they cannot be produced in vivo within humans or animals. And deficiencies lead to increased risk of certain diseases and health factors, such as cardiovascular diseases, stroke, certain cancers, and then the one we all think of course, is neurological defects within children. So the table on the right then shows recommendations for daily intake values, and it's based off of age, sex, and then of course you need higher intake values whether you are pregnant or lactating. Now I'm going to introduce you a little bit to our breeding program here at North Carolina State University. So currently cultivars are developed through conventional pedigree method with only phenotypic selection. We work with Virginia type peanuts and we focus on traits such as yield, grade, and disease resistance. Quality traits then, such as folate, are of lower priority and are only looked at later in the breeding stages, but I would argue that they are equally as important and should be considered earlier on. The official method for quantifying folate is a tri-enzymatic microbiological vitamin assay, and it's the AOAC method 960.46. And it's a very long process, and some of the reagents are hard to get, so it actually has to be outsourced at a different lab, and it costs about $142 per sample, so quite expensive to do. So the development of molecular markers for marker-assisted selection then would reduce the cost. We would be able to do it in-house and it would improve the efficiency. So the hypothesis for the study then is that folate is genetically controlled and there are molecular markers that relate to this trait, which leads to our objective to identify molecular markers associated with increased folate in cultivated peanut and may be used in marker-assisted selection. To do this then, we took our phenotypic data from the mini-core collection. The mini-core is a collection of 112 peanut lines that were selected based on an analysis of phenotypic traits. This collection should theoretically hold the genotypic variation that is seen within peanuts. Then in 2009, Dean et al. Published a paper where they looked at some of the nutritional values of the mini-core collection, one of which being folate content. As you can see from this graph, the range in the mini-core is anywhere from 102.8 to 238.1 micrograms per 100 grams fresh weight. And I'd like to point out that it's a linear trend that we are seeing within the mini-core collection. So that leads us to believe that it is a quantitative trait and there are multiple genes that are controlling folate content. Then for the genotypic data, again, with the mini-core collection, we used 2 different data sets. The first was a merged data set from the 58K and the 48K SNP array. So in 2017, the 58K array was published and they genotyped the mini-core on this array. It was then updated in 2018 for better marker positions and validation of the SNPs. So we were able to merge the 2 and use that as our first data set. And then the second came from a paper published last year. And they took the core collection of peanuts and genotyped it with the 48K array. We were then able to subset the mini-core from this data, and they were able to do some replicates as well. So we only kept consensus markers of the ones that were replicated. So for the materials and methods, we ran a genome-wide association study, or a GWAS, as I'll be referring to it as. This analysis was conducted in Tassel5, which is a platform that is used to visualize and analyze genotypic data. Both of our genotypic data sets were filtered using standard practices, which are listed here on the screen. And then the phenotypic data was the folate data that I talked about earlier from DNet et al. 2009. We used 4 subpopulations, which was consistent with the findings in Odiyama et al. 2019. And then we ran a general linear model with 1,000 permutations. Here on the screen, you will see the results of our mini-core GWASs. So for the first data set, looking at an alpha value of 0.05, there was only one permutation p-value that fell below this. And so we would only consider one of the markers to be statistically significant. We thought, though, that there could be things to be seen from any of them that had a marker p-value, though, of below 10 to the -4. So any of those are also listed. For the second data set, at the same alpha value, there were no markers that were considered statistically significant. But again, we kept any of them that had a marker p-value less than 10 to the -4. Highlighted in blue, then, you will see that there was one marker that was consistent between both studies. We then wanted to look at the position of these markers. So you will see in the second and third column, that is the progenitor positions of the markers. The next 2 columns then are the positions on the TIFF runner version 1 genome that came from the 48K array. And then in the final 2 columns, I took 150 base pairs on either side of the progenitor positions. And I used that sequence to then align to the TIFF runner version 2 genome using bowtie 2. And you'll see that most of the positions are consistent, but there are some variability between the 3 different positions shown. We then wanted to identify potential folate genes within peanut. So we identified protein sequences for the 9 enzymes that are involved in the folate biosynthesis pathway from metacago, soybean, and common bean. We then used those protein sequences to blast to the TIFF runner version 2 genome to identify peanut orthologs. And this was to determine if significant markers from the GWAS were near any of our conserved folate genes within peanut. You'll see here, this is the list of the 36 genes that we identified through the blast. But we did not see that any of the markers that were identified within the GWAS to be near any of these genes. So in summary of our results, between both GWAS, only one marker was considered significant, and one marker was consistent between both of the studies. Several other markers, though, are worth investigating further. None of our top markers from the GWAS were close to suspected peanut folate genes, but that may be because we had low statistical power for rare alleles, or it may be because folate is tightly bound within the seed and quantification may be difficult and inaccurate. So we're not sure exactly why yet. So moving forward, we have 3 folate recombinant-inbred line populations. So this was done by taking 3 of the top lines from the DNet et al. Study, and it was crossed with a really low line, a common cultivar Georgia green. The F1s then were selfed until the F7 generation, and which gave us a total of about 575 lines that we should be able to use to see variation within folate content. So the 4 parents of those populations were genotyped using 10x whole genome sequencing. from that, we were able to identify 6,766 biallelic markers with Georgia cream. And we then sequenced this using Allegro targeted genotyping platform. So we were able to prepare the libraries in our laboratory and then send them to be sequenced on a NovaSeq sequencer. And then from that, we ran a bioinformatics pipeline, which gave us 5,347 of our targeted markers. From that data, then, we are in the process of selecting one of the populations to be analyzed by Medallion Laboratories of Minneapolis, Minnesota, using that AOAC method 960.46 that I mentioned in the introduction. We are only able to do one of the 3 populations because there are a lot of lines with 576, and it's quite expensive to do. So we're trying to limit the number. And the goal of this then is to use joint linkage mapping and identify quality trait loci associated with increased folate content. So with that, these are the references that I used within my presentation. And I would like to say thank you to North Carolina State University and to the Peanut Breeding and Genetics Lab, and then all of our funding sources. And thank you to you all for listening. And I would be happy to take any questions.